It's from John J. Miles, James Cook University, Australia. The title of the presentation is Failures in Immunity are Associated with Acquiring the Emerging Disease Non-Tuberculosis Mycobacteria. Yeah, Doctor, you can start with your presentation.
Yeah, thank you for the invitation. My lab works on immunology. We work on knowledge discovery, also diagnostics, new drugs, and vaccines as well. And we are working on this project over a few years. The clinicians in our country are having a problem with this disease, so they asked for our help. So this was done by Champa, an excellent PhD student. She's now started her own immunology lab back in Sri Lanka, and she's doing quite well. So I'm in Cairns, north of Australia. We've got a brand new building. We're right next to the Great Barrier Reef and the Daintree Rainforest, which is the oldest rainforest in the world. So it's a nice place to do our work and live. I'll just quickly go over the immune system. We like to box things in, but it's gotten more complicated now. We know the adaptive immune system is around 400 million years old. The innate immune system is probably over 1 trillion years old. And with single-cell sequencing now, we're seeing lots of different types of these cells. So, for instance, T cells on what we work with, we're seeing over 150 subtypes, and they're yet to be fully analysed. So there's a lot of work to do. The difference between T cells and B cells is B cells secrete antibodies, so they're like artillery, where T cells are highly mobile and just scan tissue all day, and they can scan cells very fast. Only 3% of your T cells are found in the blood, the rest are in your organs and tissues, where they keep scanning for abnormalities, whether that's a pathogen or cancer. Now, most people are aware of tuberculosis, but most of this genus is made up of what's called non-tuberculosis mycobacterium. We know of at least 170 different NTM species. They're found in the environment. They're found everywhere, on your skin, on the desk, in the environment, in water sources. Previously, over the previous decades, they only affected people that had a problem with their lungs or if they're immune compromised. But we're now seeing a global increase, and these are more antibiotic resistant than TB. And the two largest problems are Mycobacterium abscessus and Mycobacterium avum, or MAC. So, as I said, they're found all through the environment, and they infect humans. They're found they're widely distributed in water sources, including hospital water and lakes and just the general water systems. Most diseases occur in the lung and are common in children, adults, and the elderly. Treatment is very, very harsh. It takes 18 months, and there's been several cases where patients actually died from the therapy, and a lot of cases where the patients cannot tolerate the therapy. So new avenues are needed. And the global burden of NTM disease is increasing, and we don't know why. In Australia, it's increasing at 18% each year, from 2012 to 2015. And there was a science paper a couple of years ago showing person-to-person transmission for the first time. So you can not only acquire it from the environment, you can acquire it from another person. The disease can manifest in two types: disseminated NTM infection. That's usually if you have a mutation in your immune pathways, or if you're immune deficient SCID, or if you have HIV or a cancer that takes out a key white blood cell that protects you. Most cases are pulmonary. And as I said, a risk factor is if you already have a pre-existing lung disease like bronchiectasis, COPD, cystic fibrosis, chronic aspiration, or alpha-1 antitrypsin deficiency. My real concern at the moment is how fast COPD is emerging, and who calculate that it will be the leading cause of death by 2030. So I'm quite concerned about that with NTM. There's also another subtype called Lady Windermere syndrome, where NTM attacks these types of people. They're older females, and they have a low BMI, and they're quite thin. But other risk factors are vitamin D deficiency, ABPA infection, and a lot of people are now on anti-TNF therapies, monoclonals. TNF is essential to clear mycobacteria. So a lot of people are getting NTMs because they're on that therapy, and also steroid therapy, which basically is a sledgehammer to your immune system. So they're the host factors, but there are other factors as well, like natural. Where they're found, we're still investigating. They're definitely in the soil, and there's animal reservoirs, there's water reservoirs, lakes, and man-made. They're quite common in showers. There was a piece in Nature a few years ago saying it's a hot tub disease, so they're present in hot tubs. Gardening puts you at risk, and they're found in hospitals. They're very well protected against antibiotics and disinfectants. They're aerosol-borne. They're very hard to identify because culture takes usually weeks, and you're not really sure if the person still has NTM. It could be in another part of the lung, and you need special media to grow it. So we need better diagnostics as well. This is just a little projection we did from our state, and we're going to see probably about a 400% increase in cases over the next 20 years at the minimum. It'll probably be higher. This hasn't factored in age. And my state in Queensland is bizarrely the only state that NTM is a reportable disease. And most countries are the same. So we can't get great numbers, and they're not found on the Lancet disease website. But yeah, they are increasing. Some epidemiology data has come out and shown risk factors are increased the closer you are to the equator. So again, that's bad news, because we know the Tropic of Cancer and the Tropic of Capricorn are increasing in length, I think about 50 kilometres a year. So that covers a large portion of Earth, and rapidly populating countries like Southeast Asia, India, Africa, and South America. So I'll first talk about Mycobacterium abscessus infection, where we had two patient groups. First patient group had cystic fibrosis, and the second patient group seemed to be perfectly healthy, but they developed NTM. Now, NTM in cystic fibrosis is a virtual death sentence. So it's very important that we try and help this population. So we had different groups. We had patients that had active disease. We had patients that had NTM in the past, and we had patients that had never had NTM. And in the middle-aged cohort, we had people that had active NTM, and we had healthy controls because cystic fibrosis affects young people, and middle-aged elderly patients have a different immune system. So it's very important to match up age and sex when analysing the immune system. Okay, so this is cystic fibrosis data, and this is done in SPICE on FlowJo. And so you can have 1, 2, 3, or 4 functions, and the arcs are what are found. So you can see the CF controls who've never had NTM and the healthy controls look much the same. And interestingly, the CF patients that have active disease and past disease are different. And the biggest difference is in TNF production. So CF patients can basically be drawn in half into patients that are susceptible to NTM and patients that aren't. And you can see with the various dot plots here that the controls pretty much match. The CF controls pretty much match healthy individuals. And whether you have an active infection or past infection of NTM, it doesn't seem to change. So my guess is these people have defects in their underlying immune setup that makes them predisposed to NTM infection. Here's just a heat map. The main thing you probably want to look at is, again, the people with either active or past NTM cluster together in the red and blue, and the CF patients who've never had NTM and in the healthy clusters, they cluster separately as well. So CF can probably be split in half into these two groups. The second group you looked at are these emerging cohorts that are alarming clinicians, where 56-year-olds are developing NTM and they don't have any pre-existing lung conditions. And so we did it again with active infection and then a different healthy control cohort that were older. So in this case, it was different. So you can see with TNF here, the blue bar, the patients with active condition are producing plenty of TNF, so that's not the problem. And yeah, much more than healthy controls. So that wasn't their deficiency, but their deficiency was in how exhausted their T cells were. So they often presented the exhaustion markers, PD-1, CTLA-4, and they were highly activated. And they also had a lot of FOXP3 activated Tregs, which further suppress the immune system. So there's two different mechanisms based on these two different cohorts. So just to summarize the two settings, in the young, it's similar across the disease groups. Their failure seems to be in TNF production, which is interesting because we got samples from the blood and the diseases in the lung, which means that we can get information quite easily just by a simple blood draw. Their capacity for interferon gamma is elevated, but you really need TNF alpha for mycobacterial control. and they had elevated exhaustion markers in their Tregs as well. So their Tregs weren't working properly, but similar PD-1 expression. In the older group, TNF was fine, no change in interferon gamma, but their cells were tired, both Tregs and memory T cells. So then we expanded the study to the other problem bacterium, Mycobacterium avium complex, or MAC. And we increased the patient numbers, and we increased the profiling. So we did flow cytometry, transcriptomics, and we're doing multi-parametric analysis at the moment. And we're also looking at checkpoint inhibitors as well. And so we had a 40-color flow panel. This flow panel basically just covered all the immune cells with exhaustion markers and activation markers. The second panel covered the non-conventional cells like gamma delta cells and mate cells. And the third panel covered various subtypes of T cells and cytokines and chemokines that they produce. So this is what you get. This is stacked. So all donors are stacked on one, and this is a t-SNE analysis. So it characterizes the cells based on what they express. So you can see all the cells have separated here. Most of the cells in white blood cells are CD4 naive. You can see everything else here, B cells, NK cells. You've got CD8 cells in different spots. You've got NK cells, again, CD4s in different spots, naive. And we have this unusual population, which we're not sure of yet, that are CD3 negative and also CD14 negative, so they're not T cells or myeloid cells. So what you can do with this is overlay each of the functional markers, and it's an unbiased analysis, and it shows you where the differences are. So here are the healthy controls, and here's our panel. These are all exhaustion markers. So this is what it looks like normally. For MAC, you can see there's a little bit of exhaustion across here, across CD4 cells. There's a heap of exhaustion in NK cells, which we weren't expecting. And in PD-1, there's exhaustion in the TEMRAs. For MABs, it's different. They don't have a lot of NK cells, and there's a lot of exhaustion in this unknown population, and also B cells. You can do further clustering of t-SNE and divide those population into nodes. And if you have enough people, you can do stats as well. And so everything that's purple here is significant. So you can see up here, these are the CD8 cells. So they're significant when you're looking at TIM3, and NK cells are significant as well when looking at TIM3. I was expecting more significance with PD-1, but we've only just got one dot there. So this is done on what's called Citrus, which is based on Cytobank, which was originally designed for mass spec, but we've rejigged it for fax analysis. And you can see with the NK cells, we're getting pretty strong significance with upregulation of TIM3 with active infection. And that's the same in T cells as well. So that might be a target for this population. The other thing we've been working with is something called nanostring. It's fairly new, but it's the first digital fully quantitative platform for gene expression, but you can use it for protein expression as well. It's basically like a barcode system, like traffic lights, and you design your probes based on your favorite genes or proteins. And what I like about it is it's PCR free, so there's no bias in amplification. It basically just counts molecules. It's over 800 plex, but I think they've added another molecule so you can get a 60,000 if you want to. And because there's no PCR involved, you don't have to worry about RNA extraction or what type of material you're putting in. And you don't need much, just a microliter. So, a microliter of saliva or blood or plasma or just any fluid. You can put into these cartridges, which hold 12. And it's just really easy to use. So you just put the cartridge in the machine, press go. After about an hour, it's finished. And you put it into the analysis machine, which has like a 40 megapixel camera. And it just basically counts the dots because they're laid flat. So all you get is just numbers of the different genes. So this is now TGA approved, and it's approved in lots of other countries for clinical diagnostics, and it's very easy to use. So nurses can use it and things like that. So we used it for this study and found something quite interesting. So you probably can't see the column, but the first column is people with active MAC. This column is people after treatment, so they're cured of it. This column is active MABs, so it's a different species. And this column is persistent infection from MAC. So a lot of patients aren't cured and they go on have persistent infection and often pass away. Now, what was really interesting is you can see, so this is compared to the healthy controls, and so we've got like 20 or 30 patients in here, and I've colored it by p-value. So the darker it is, the stronger the correlation. So blue is down compared to healthy controls, and red is up compared to healthy controls. And I wasn't allowed to name the sorry about that. I wasn't allowed to name the genes because business development said I couldn't. But they are immune genes, and what you can see is basically different footprints. So when you're actively infection, a lot of genes are turned down and a lot of genes are turned up. These sort of disappear after treatment and the pathogen's gone. You get a different pattern with the different species, which I wasn't expecting. And also persistent infection is almost no different to healthy controls. So it looks like in this cohort, their immune system is not aware of the danger in the lung, and they're not doing anything about it. We were also wondering if you could even tell the difference between the species, even though they're the same genus. And it looks like you can. So these are the different genes, and you can get a pattern. And so, yeah, from a blood draw, and just a couple of hours, you can tell what species a patient is infected with, rather than a couple of weeks from a 40-year-old culture system. And so, yeah, here are the p-values, and some of them are very, very strong. And so we're doing a pattern analysis now and a UMAP to get a signature. But yeah, so you got forest plots, so you got genes that are upregulated and downregulated, and we've got good FDRs and AUCs and p-values. And you can use LOCV, and that can identify a signature. And looking at T cells, we can detect these infections with 95% accuracy, 100% specificity, sensitivity, and 90% specificity. And for the CD8 cells, 91% accuracy, 80% sensitivity, and 100% specificity. So that's a huge advance on what's available now, but we have to upscale. So my vision is pinprick of blood in the nanostring or a lab on the chip. We're looking at nanopore at the moment, which is just a dongle that you can put into your laptop, and you can get data within seconds. So that's the path we're taking. And to summarize, there are a few papers coming out on this on different diseases, that your immune system can be a proxy diagnostic for the disease you have, because T cells are continually circulating and we think they're talking to each other. My nickname for it is chatter. So, you know, your little toe knows what's going on in your spleen or heart or lung, and even possibly the exact threat. And as I've shown, they can be very sensitive and specific and accurate, and also rapid. And they can be used to guide clinical decisions. So if we get feedback saying that a patient is probably going to be persistent, the doctors can alter the drug course. Also, if we identify people who are at risk of this disease, they can change their lifestyle and start having baths instead of showers, and, you know, ask them to keep out of the garden, that kind of thing. And yeah, we're going to go down the rabbit hole and just try and figure out what is going wrong so we can fix them. Like, for instance, in cystic fibrosis, we might be able to add TNF therapy to help them control disease. So yeah, to future directions, we want to increase the cohorts from dozens to hundreds, and multi-site across the world to make sure it's solid. Yeah, just through validation. And yeah, we're going to follow up the immune genes for mode of action, so we can see which pathways are defective, and if we can either redeploy drugs or make new drugs to correct the dysfunction. So almost all this work was done by Champa, so she was just excellent. But I'd like to thank everyone in my lab and our clinical collaborators that provided the samples, and our funding sources as well. So yeah, thanks for listening. And
thank you for your presentation, Dr. John Miles. It was very informative.